So at some point in time, you were just a single cell. No thoughts, no memories, just a tiny self-contained package of biological material. And somehow that cell knew exactly what to do. It divided and divided again until in a matter of weeks it had formed the blueprint of a brain, some limbs, and a beating heart. Now, incredibly, there is no tiny architect sitting inside the cell directing its actions, no grand plan being read aloud as these cells make decisions. Instead, each cell is simply following internal rules, listening to the chemical whispers of its neighbours, and making decisions based on the decisions that it's made in the past. And yet, from this apparent chaos emerges something incredibly organised and ordered, each of us. So today I want to take us on a journey through this process of embryo development, this incredible choreography that establishes the body plan, where cells negotiate, compete, cooperate, and sometimes make mistakes, and yet, more often than not, we end up with organs and tissues formed in the right place and at the right time. Because if we can understand something as complex as how the human body forms from seemingly nothing, then we can maybe understand something even bigger, how simple rules can lead to extraordinary complexity. I'm Dr. Naomi Moris. I did most of my training at the University of Cambridge, and I now run a research group here at the Crick called the Developmental Models Laboratory. So humans have been fascinated by the process of development, where we come from, since time immemorial. This is one of my favourite drawings by Leonardo da Vinci, who depicted the human fetus inside the womb in a way that I think looks a bit like a conker inside a shell. And it is an incredible process, right? We go from essentially a single fertilised egg cell through the process of cell division. So a cell divides in half to become 2 cells, which divide again to become 4 cells and 8 cells and so on. And then those cells interact with one another in order to form the organisation and the pattern of the body structure. So let's take a step back for a minute and think, if I were to hand you right now a lump of clay or a piece of Play-Doh, how would you go about building a body? Would you perhaps start off with a very simple structure and then start to add in complexity as you went on? Or would you maybe start to sort of generate discrete parts and put them together, an arm here and a head here, and start to assemble? Essentially, the embryo is doing both of these things, but it has an added challenge, right? I've handed you the clay or the Play-Doh. The embryo actually doesn't have all the starting material it needs. It has to literally grow the cells as the cells are making decisions and as this organisation is forming. Now, this is a kind of perplexing situation, right? And people have been confused about this idea of something arising from nothing for a very long time. And in fact, this is a drawing from the 17th century by Hartsucker, where he was so puzzled by this question of how something can come from nothing that actually he reasoned that inside the head of a sperm must be sitting a little tiny person called a homunculus that would then just simply grow during development. It's kind of an attractive proposition, right? Except that it becomes a bit silly if we think this might be a baby boy who would then also have to have sperm inside him, which would also contain little tiny people, and they would have to have little tiny sperm with little tiny people. So it becomes a bit ridiculous. And so we now know that actually this process of development is not just growth. Actually, it's a process that's incredibly dynamic, and it's a process that is very progressive. So development is changing these cells over time as they divide. They're also establishing pattern, and they're being molded into different shapes that allow the structure of the body to form. And in fact, most of the process of development is kind of established or laid down in the very first weeks of embryogenesis. So up until 8 weeks of development, the structure is called an embryo, and that essentially sets up the body plan, the organisation. And beyond that point, we call the structure a fetus, and this is mostly kind of growth and a maturation of the organs that are already present. So in this talk, I'll focus mostly on the embryo stages, because that's the stage of development where a lot of this complexity is unravelled. Okay, so let's walk through the stages of embryo development slowly, hand by hand. So of course, development starts off with fertilization, sperm meets egg, and the 2 pronuclei of those sperm cell and the egg cell fuse together to form a new genetic individual. After this point, when we've got essentially a single fertilized egg cell, that cell will divide, which means the cell splits in half to form 2 new cells, and those 2 cells split in half to form 4, which split to become 8 and 16 and so on. And you can see here that at the outside of this structure, which looks a bit like a blackberry, if you like, with these lots of different cells put together, you can see this jelly-like layer around the outside, which is called the zona pellucida. And this is essentially a layer of material that protects the early embryo during these early stages of development. So as the embryo carries on development, it starts to distinguish between the cells that are on the outside of the embryo and the cells that are on the inside of the embryo. And it actually forms this fluid-filled cavity in the middle. So it's a bit like a water balloon or a bubble, with cells around the outside, this fluid in the middle, and then a few cells that are inside the structure. And this is called the blastocyst, which is an incredibly important stage of development that we'll be coming back to later. Then the blastocyst actually hatches. So you're probably aware that chickens hatch from eggs. We also hatch during our developmental process. And what we hatch from is the zona pellucida, so this jelly-like layer. The blastocyst hatches from that zona pellucida. And that's really important to allow the embryo to do the next stage of development, which is implantation. So once it's hatched from this jelly layer, it can then implant into the endometrium, which is the lining of the uterus, to allow the embryo to carry on its development in interaction with the maternal environment. So after implantation, the embryo starts to rearrange and change shape. And one of the first things that happens is that it forms various cavities. So one of these is the amniotic cavity, which forms the amnion, this layer of cells that cover the early embryo and protect it. And we can look at the actual embryo itself, the part of this structure that's going to form the actual body plan at this time point, as just this disc of cells, so this thicker region here in this picture here. That's the only part that's actually going to form the body of the structure. All of the other tissues are what we call extra-embryonic tissues. They're the structures that are going to support the growth of the baby, so things like the placenta and the yolk sac that help the development of the early embryo. So this is a side-on view of that disc of cells. You can see here this thicker layer of black cells here in this picture. If we instead take a top-down view of the embryo at a slightly later stage, we can see this crucial moment of development called gastrulation. Now, I know that's a scary word. Gastrulation just means that it's basically when the embryo starts to determine which side of it is going to become the head and which side of it is going to become the tail. And you can tell the tail end of the embryo because I hope you can see that there's this fine line that exists along the embryo at this, where these arrows are pointing, which is called the primitive streak. So that part of the embryo is now going to become the tail end of the embryo, and the topper part is going to become the head. After this point, there's a lot of rearrangement. The embryo starts to elongate along this head-to-tail axis. We start to see the morphology, the shape appearing as the embryo starts to establish all the different tissues that it needs and the early organs. And essentially, this is a kind of remodeling process as the embryo takes shape. So how do we find out about development? How do we study this process of embryo development? And particularly, how do we study mammalian embryo development, where, of course, the process is happening inside the womb? One way that we can study this process is actually by using other species. So the mouse has been an incredibly useful model system to be able to understand mammalian development. So let's take a breather here and do a quick straw poll. I've put up here 2 pictures of embryos. One of them is a mouse and one of them is a human at essentially an equivalent stage of development. Let's raise a hand if you think the one on the left is a mouse. Oh, nervous hands. Some, okay. And who thinks the one on the left is a human? Oh, more hands. Okay, interesting. It's not that far off, right? It was not too far from 60-40 as a kind of percentage, right? And I think that means that it's a difficult decision, right? It's hard to know which one is a mouse and a human. Here you go. This is the one on the left was a human, the one on the right was a mouse, and I've made it harder because I've hidden the tail. So the tail part of the embryo would have given away that it was a mouse, and it's obviously behind the structure here. But the point of this slide is really to say that the mouse and the human, in some aspects, are quite similar. So there's a lot that we can learn about human development from looking at the way that the mouse develops. The other way that we can learn about human development specifically is from processes including in vitro fertilization, so IVF. Now, this has been an incredible advance that's been used by millions of parents around the world who want some help to conceive. And it's been really a revolution in our ability to do assisted reproduction and lead to successful pregnancies. But it's also been a revolution in our ability to do human developmental biology and research into the early human embryo. Because if couples have surplus embryos or surplus eggs and sperm, they can choose to donate that to research to allow scientists to undergo study into these earlier stages of development. What that means, importantly, is that we might be able to better understand how to improve the conditions of IVF to lead to more successful pregnancy outcomes for these couples. And this important scientific research is only possible because of their generosity in donating this material, and it's already revealing a lot that we didn't already know about human development. The other way that we can learn about human development specifically is from terminations. And so these, again, are generously donated by patients. And in fact, there are several very large collections of these embryos across the world. So this is an example from the USA called the Carnegie Collection, which allows us to essentially study the embryo at later stages of development and be able to draw up classifications of different stages of embryogenesis. So we can look at early embryos and later embryos and see exactly how the embryo is changing over time. But these two kind of sources of information lead us to a bit of a problem that's sometimes called the black box of human development. So in the UK, you're only legally allowed to study human embryos under a license and only up until day 14, which means that we can only use IVF-derived embryos to research that first 2 weeks of human development. The collections that exist and the access to samples from terminations are very rare to get access to before 6 weeks of development, and even rarer before 4 weeks of development. So this means there's this kind of gap in our knowledge where between weeks 2 and weeks 4, we really have no access to actual embryos to be able to study this process of human development. And yet that's the key time point when we start to see the body plan forming and the emergence of early organs. So it's a crucial window to be able to understand. Which leads me to my second part of the talk, which is how can we instead use stem cells to be able to model this time window of development? So stem cells, pluripotent stem cells in particular, are sometimes called the building blocks or the Lego bricks of the embryo, because they're essentially the cells that make up this early embryo when it's starting to make decisions. So pluri just means many, and potent or potential just means the ability to make many cells. So these cells are kind of like the superheroes of the dish, right? They have these two powers. They can both make any cell type of the body, so they can be turned into skin cells or brain cells or heart cells or muscle cells, any cell type of the body. And they can also self-renew indefinitely, which means we can keep them growing in culture, make millions of them, and keep them going essentially indefinitely. Which means that they're an incredibly powerful way to unpick how cells are making decisions by using them in the lab. So there are two ways that we can get pluripotent stem cells, one of which is to take them from a very early embryo. So an embryo at the blastocyst stage, when it's this kind of fluid-filled bubble, we can take the cells from the inside of that structure and grow them on a dish in the lab. And they have these two properties, right? They're able to differentiate into any cell type, and they're also able to self-renew indefinitely in culture. The other way that we can make these pluripotent stem cells is a method called induced pluripotent stem cells. And this is essentially where we take a sample from a donor or a patient. It could be a skin biopsy, it could be a hair follicle, it could be a blood sample. And then we can reprogram those cells back to an embryo-like state just by throwing on a handful of genes, which is an incredible advance. It won the Nobel Prize. This means that we can essentially take an adult cell and make it go back in time to something that's a bit like an early embryonic cell. And then both of these cell types, these embryonic stem cells and these induced pluripotent stem cells, are essentially equivalent, right? They both have this capacity of self-renewal, and they both have this capacity of differentiating into any cell type of interest. And what has recently been discovered about stem cells, both pluripotent cells like I've described here, but also adult stem cells, is that they have this remarkable capacity that if you put them together as an ensemble, as an aggregate, they start to have properties that we wouldn't otherwise expect. So they're able to communicate with one another and interact with one another in the same way as they would do in an organ or in an early embryo, which is an incredible feat, right? This idea that they're able to interact and essentially what's called self-organize. Now, self-organization is not a new concept. It's not something that's specific to biology. It's found throughout the natural world. It's found in physics and chemistry as well. And it's essentially this idea that if you take discrete parts or individuals and allow them to follow local rules and interact with their neighbors, you can actually end up with something that is incredibly complex as a kind of emergent phenomena. So here we've got some birds that are acting in a murmuration. This is a beautiful display, but essentially each bird is just following its neighbors and interacting with things like the winds that are acting as a cue to form these really beautiful organizations of structures that just emerge from these simple rules over time. The same thing is true of cells, right? When you put cells together, they can interact in ways that we wouldn't otherwise predict to lead to really complex outcomes. And so we can leverage this fact to make what are called models in the lab. And essentially what this means is that we take the cells that we would normally grow on flat plastic dishes, and they're usually cultured in this fluid, which we call media, which has all of the sugars and the growth factors and the chemicals that the cells need to keep them alive and to keep them happy. And we take those cells that are normally adhered on, so stuck down onto this flat plastic dish, and we can use enzymes to separate them out gently and cause them to be single cells, individual cells, a defined number in a concentration in this fluid that we grow. And then by replating an exact kind of defined number of cells into each well of one of these 96 well plates, we can ensure that these cells are kind of coming together within a range of kind of numbers and watch as they interact with one another. And so because these cells are really sticky, they'll essentially stick together, form a little spherical structure first, and then start interacting with one another in order to set up different regions of this structure with different identities. So here we've used some fluorescent reporters to basically explain which cells are expressing which genes, so be able to visualize that down a microscope. And we can use that to see how this structure has now gone from a spherical structure to something that's elongated, and that these colors you can see are in different regions of this structure, which means that the cells have organized themselves relative to each other. What this means then is that we can tweak this protocol in different ways. So we can mix different types of cells, we can use different types of media, and by doing that in the right kind of defined conditions, we can actually lead to different outcomes. So this allows us to kind of tweak these protocols and uncover what these cells are capable of doing, but just in a dish. And so this has led to this new field that is collectively called stem cell-based embryo models. So stem cell based because they've come from stem cells, and embryo models because they allow us to model some of the features of an early embryo. Now it's a really broad group of structures. I've shown you a few examples here. There are lots of different flavors and different ways that we can generate them, and they each have different pros and cons, different advantages and different advantages. So I like to see them as a bit of a kind of toolbox, if you like. We can pick the right model to answer a particular question. So let's have a look at 3 different examples of questions we might want to answer using an embryo model. So firstly, maybe we want to investigate how implantation happens, right? So we don't know a huge amount about how the embryo actually interacts with the maternal environment. How does it know when it's ready to interact with the endometrium? How does it actually do that sort of sticking in the first part to the endometrium? And how does it know which way round to do that, right? These are all questions that we actually know very little about because, of course, they happen in the womb. And so we can use these embryo models that are called blastoids. They're potentially the most famous of the embryo models, which, as the name would suggest, oid just means similar to, blast means they're similar to the blastocyst stage. So essentially they look like this. They've got this kind of large bubble structure with these cells around the outside and then cells on the inside. And they really model this early stage of development before implantation happens. That means then that they're made of these extra embryonic tissues and the embryonic tissues. And because they're made from stem cells, we can make them in huge numbers to be able to look at different chemicals and their effect on the early embryo. Now, it's important to state that these blastoids, although they look fairly indistinguishable from actual sperm and egg-derived embryos at the blastocyst stage, even if you make a mouse blastoid and put it back into a mouse uterus, it's never given rise to a fetus, and let alone live offspring. So they look very similar, we can learn a lot from them, but they're not equivalent to a sperm and egg-derived embryo. But what it does allow us to do, as I mentioned, is to be able to begin to unpick how implantation might happen, at least the early stages. So here you can see these researchers have made these human blastoids, and they're culturing them on this layer of endometrial cells. And you can see as they're kind of wobbling these structures with their pipettes that actually they're stuck down. So these structures have started to actually interact with this maternal environment, and we can use the system to unpick exactly how that's happening and what these kind of signals are that happen between the cells. The other thing that we might be able to use models to understand better is these interactions between the embryo itself, the embryo proper that gives rise to the body plan, and these extra embryonic tissues that are really important for supporting the early stages of embryo development. So these are a kind of range of models known as post-implantation embryoids, and they essentially skip this first part of development and go straight to the post-implantation stage of development. And yet they allow us to explore these interactions between the embryo itself and the supporting tissues. What exactly are those chemical whispers that are happening between these cells, and how is that influencing how these actual embryo cells are making decisions? The third example I want to give you of an embryo model and the kind of questions we can answer with it are called gastruloids. And these, as the name would suggest, the oid means similar to, gastro means similar to gastrulation stage. These model this time of development when the cells are starting to decide which cell type to become. So whether to become the muscle cells and the brain cells and all other cell types of the body. Now, these embryo models, these gastruloids, allow us to unpick exactly how these cells are making decisions and how the gene expression is organized along the anterior to posterior axis, so the head to the tail part of the embryo. And that's because they have this elongated structure that mirrors this head to tail axis of the early embryo. So one really nice example of this is a gene signature called a Hox genes. And this is an incredible set of genes, right? These are genes that are ordered along the DNA sequence from number 1 to number 13. So a bit like beads on a string, 1 all the way through to 13. And what we think happens is that these genes start to open up sequentially, one at a time, as the embryo is developing, which means that the earliest Hox genes, number 1 and 2 and 3, are expressed in the head region of the embryo, and then the later Hox genes, as these genes open up, start to become expressed in the tail part of the embryo. And so the order of the genes along the DNA sequence matches both the spatial localization of the gene expression, but also the temporal localization of gene expression, so when they're expressed over time. And we can use this gastruloid system to start to explore how these Hox genes are regulated. So if you use a gastruloid at an early time point, you can see gene expression, so the opening up of just those early genes, and through later expression, we get the opening up of some of the later genes. So maybe we can use the system to explore how gene expression is regulated and how the embryo patterns its head to its tail. Okay, so I've given you a broad introduction to embryo models and a flavour of some of the questions that we can answer with these models. So now let's do a bit of a deep dive into some of the questions that we're asking in my lab using these embryo models. And so the first little story I want to tell you is about my favourite cell type, and these are called the germ cells. Now, these are incredible cells. They're cells that go on to form either the sperm or the egg. And they're specified actually really early in development. So the cells are set aside, ready to make either sperm or egg as the embryo is developing. And what's incredible about them is that actually they essentially kind of go through this process through development and then give rise back to the sperm and egg, which of course confuse again and lead to another embryo development. So as I said, they're specified early. They then move through the gut tube, which is a kind of weird way to travel, right? They move through the gut tube from this tail end of the embryo up to what are called the gonads, which is either the ovary or the testis, depending on whether the embryo is female or male. And then the gonads will tell these cells, right, you are in a female embryo, I need you to make an egg. Or you are in a male embryo, I need you to make some sperm. So it's an incredible process. It's really interesting that these cells are set aside early, and we really need to understand these cells because they're the ones that are crucial for passing on the DNA to the next generation. So they're a really key cell type. And one of the things we found out is that actually you can use embryo models to start to study these early germ cells. So here we're using mouse gastruloids, so made from mouse pluripotent stem cells. And we can see that these structures start to specify early germ cells at around the same sort of similar time to the early embryo. They then associate with the endoderm, so this gut tube structure that exists in the embryo. Then they migrate or have elements of kind of migration that looks like the cells are moving. And then they start to form these clusters of cells that exist in these gastruloid structures in a similar way as they would do in the mouse embryo. Now this is incredible actually because this whole process involves so many other tissues, right? These cells have got to travel, they don't just stay in one place. They interact with a whole sort of journey of cells along the way. And this is an embryo model that allows us to explore each of those interactions in turn and see how it affects these germ cells. The other thing that I think is really interesting is that the classical way of making germ cells in a dish is to make what are called embryoid bodies. And these are large aggregates of pluripotent stem cells that we then give a cocktail of chemicals to push them in a particular direction. So we might push them kind of chemically towards a particular lineage or other. Now this is an incredibly efficient way to make germ cells. You can make lots and lots of germ cells with this method, but because you've pushed them in a particular direction, you're not actually watching what those cells would normally do in an embryo and how they would normally interact with their environment. And we showed a couple of years ago now that actually if you compare germ cells that are made with this traditional method where you're pushing cells towards making germ cells, you actually get quite early or immature germ cell outcome. But if you use an embryo model approach, so this gastruloid approach, you can actually get more mature germ cells. So we think this means that actually the environment that the cell experiences, this embryo-like environment, is really important for the maturation and the fidelity of the cells we get at the end. The second story that I want to tell you is a bit about this interaction between tissues, right? These chemical whispers that happen as cells are developing in the embryo. And to do this, we have to look at your back, right? So your back has a spine, which has these kind of vertebral bones that sit along the back part of your back. And they're kind of repeated segmental blocks of bone, right? And that structure actually appears very early on in embryo development. So this is a human embryo, and you can see the kind of outline of the structures here. This long tube that runs along the middle of the structure is called the neural tube. That will go on to form your spinal cord. That's got all of the nerves of the kind of posterior part of the body. And these blocks of tissue on either side, these repeated segmented blocks, are called the somites. And they're going to go on to give rise to these vertebral bones, but also some of the musculature of the body. Now, if I take a cross-section, so if I slice this embryo in half here and have a look at it through the middle, you can see this neural tube outlined here in blue sat right next to these two green somites. And so what we think is that there's probably interactions that are happening between the somites and the neural tube that are really important for this embryonic process of development to occur. But of course, this has been really hard to study in actual human embryos. And so what my lab did last year actually now is to start to make an in vitro embryo model system, so a system made out of stem cells that allows us to study this neural tube and these blocks of tissue called somites, but in a dish. And so we generate these structures that have both a tail end here in pink, but also this neural tube structure in yellow and these blocks of tissue that are called somites here in cyan. And what's great about this is because we make them in the lab, we can actually watch as they develop. So here you can see this gene that is becoming expressed and then not expressed, it's oscillating. And as it oscillates, it starts to form these blocks of tissue called somites that are appearing from the anterior part, the top part of the structure towards the bottom part. And so one of the things that we're able to find with this kind of model is exactly how these tissues are communicating. And so we think actually what's happening is that there are signals that are coming from the somite that pattern the neural tube, but there are also signals coming from the neural tube that pattern the somites. And one of the ones that we identified was a signal called retinoic acid, or RA, which comes from the somite and signals back to the neural tube. But we noticed when we looked in our in vitro cultures that actually one of the genes that's really important for regulating retinoic acid, called ALDH1A2, was not expressed throughout the whole somite. It's only expressed in the part of the somite that's touching the neural tube. And so that implied to us that maybe there's a signal coming from the neural tube that's also patterning the somite. Now, that had never been described in embryos before, and so we were able to go back to the mouse embryo and check exactly where this gene was expressed in the embryo. And here we saw again that this gene was expressed only in the part of the somites that was right next to the neural tube, which I think tells us that actually we can use these embryo models to start to learn something about the embryo that we didn't already know before, which is a really powerful way of understanding developmental biology. Okay, so we've gone through a lot of the science. Let's think a little bit about what the applications of this kind of technology could be and what the future might hold for this kind of research. So I hope I've explained to you that there is a really important fundamental understanding of human development that we can only really gain access to from using these kinds of embryo models. But there are also other advantages to using this kind of system. Not only might it allow us to replace, reduce and refine our use of animals in the lab, but it also, because we can make them from induced pluripotent stem cells, might mean that we can represent more diversity by looking across different genes and populations to be able to see what their effect is in early development. There are also a range of biomedical applications that we're going to run through in a minute, including the possibility of improving IVF outcomes, doing drug screening, and looking at environmental susceptibility of the embryo to outside exposure risk. We can also start to look at development of diseases and other conditions that occur in early development, and maybe even produce some of these rare cell types that are otherwise really difficult to generate in the lab by leveraging the power of the early embryo. So let's have a look at these in a bit more detail. So one of the things I said we might be able to do with these embryo models is potentially to improve IVF success rates, which is incredibly inefficient at the moment. And so by using things like these blastoid models, because we can generate thousands or tens of thousands of them in the lab, we can essentially use them to look for conditions, for chemicals that we can use to support their growth, to allow them to grow really well, and to enable them to get to kind of later stage development. Now, this might allow us to identify exactly the composition that we need to use to grow our actual sperm and egg-derived embryos. So perhaps we can identify a better media composition for actual IVF-derived embryos to enable more live, successful pregnancies. The other thing that we might be able to do with embryo models is, because we can make them in large numbers, is use them for screening different drugs. So in particular, we know that during pregnancy, you're susceptible to exposure from various compounds and chemicals that can affect the developing embryo and developing fetus. And so we'd really like to be able to predict, well, which of these drugs is going to have an effect on the developing embryo and which of them are potentially safe for use. So it's early days for this sphere of research, but we've done a little bit of kind of proof of concept of this, that we can use these gastroloids made from both mouse and human cells to test known teratogens. So these are drugs that we know definitely cause effects in early embryogenesis, and have a look at how different concentrations of exposure to that drug can lead to diverse outcomes in this embryo model. The other thing that we can do is to look at drugs that might have a specific human effect. And I should say at this point that this is potentially a sensitive image, but this, obviously, the most famous case of a drug causing some sort of these effects is thalidomide, which of course was marketed as an anti-morning sickness pill and therefore given during early pregnancy. And it led to these really devastating limb defects, but it also led to a lot of miscarriages in early development. And the reason that it was allowed to be released onto the market is because actually it has almost no effect on mice. So it was tested on mice beforehand, had no effect, and therefore released to humans, assuming it would be safe. And of course, it wasn't. And so we're able to use embryo models made from different species to be able to hopefully predict whether any of these effects are going to be human-specific, right? Is there any benefit to using a human cell model to allow us to predict the effects of these drugs? And indeed, in the case of thalidomide, it looks like the human gastroloids are having more of an effect than the mouse ones. The other thing that we can do is that early development is very susceptible to any kind of genetic changes that can cause these what are called congenital abnormalities, conditions at birth. And so this is an example of a condition called spondylocostal dysostosis, which occurs when there's a mutation in this HES7 gene. And you can see this is the vertebrae of a patient. You can see that the vertebrae are completely misformed and misaligned, and they lead to this really kind of disorganized pattern along the spine. And so we're able to use embryo models to say, well, what is the kind of healthy effect of this process of somatogenesis, where this is thought to arise? And then can we make the same structures out of cells that have a mutation or a loss of that particular gene, HES7? And you can see from these examples here that actually we've completely lost that blocks of somite epithelialization, those blocks of tissue that form these early vertebrae. We can also potentially use these embryo model systems to understand other congenital abnormalities. And this is a baby who had spina bifida and had surgery to correct it. And of course, spina bifida and other neural tube defects are actually relatively common. And although there are excellent surgical treatments now for spina bifida, one of the things that we know is really important is the addition of folic acid or folate into the maternal diet, right? So supplementing with that nutrient actually minimizes the risk of these conditions like spina bifida. It's entirely possible that there are other chemicals or other nutrients that we should also be supplementing the maternal diet with that might therefore reduce the incidence of some of these conditions, and we just don't know it yet. So hopefully embryo models might allow us to understand or to even potentially find new treatments for some of these conditions. So I've told you a lot about some of the benefits of these embryo models, but of course the flip side to this is that they raise a lot of ethical conundrums, right? And so we're generating something that is made from human stem cells that mimics the early stages of human embryo development. And there are a lot of unanswered questions about what we think might be worth protecting or worth preventing when we're thinking about generating these models. So some of these pre-implantation models like the blastoids, could these ever be used to create a live fetus? And if that happens in the mouse, would we then be worried about the same potential existing in the human blastoid? And because they're so similar to the embryo, how would we ever know that we'd kind of reached a tipping point that these two things were now equivalent? So I said when I introduced blastoids that they're not equivalent at the moment, but would we really be able to tell when that happened and they became essentially equivalent to one another? And therefore, should we be allowed to grow these stem cell-based embryo models beyond day 14? That's our limit for growing sperm and egg-derived embryos. Should we have the same limits in place for something that's just made from stem cells? The same sorts of questions are true for some of these post-implantation models, right? I've told you that they skip these earlier stages of development. Is that important ethically? Does that make a difference to how we feel about these structures? And most experimenters have kind of stopped a lot of these experiments at about day 14, so the limit for actual sperm and egg-derived embryos. But should they have to? Should we be able to culture them a bit further if it gives us insights into early human development and some of these conditions that have obviously are caused slightly later on in development? And for some of the post-gastrulation models, so things like the gastruloids and other things, are there specific features we'd be worried about seeing in the dish? Would we be worried if we saw a structure with a beating heart or limb buds or skin or whichever other organs? Should there be a limit on how complex these things can get in a dish or how kind of long we can culture them for? And these are very much unanswered questions. I don't have an answer for you, and I've been thinking about it for the last 10 years. And so, we really just don't have a good ethical understanding of some of these questions, and I think it's an open discussion for us all to have where we think these lines should be drawn. Which leads us to a bit of a governance gap. So obviously these things are not embryos. They're not the same as a sperm and egg-derived embryo. And so the same rules that apply to embryos just can't really be applied to embryo models. They skip stages. They don't necessarily mirror the whole embryo. They have different capacities and potentials. So we cannot use the same regulation as we use for embryos for embryo models, but that leaves us with a bit of a gap. So what should we regulate? How should we regulate these things? Who should regulate these things? It's still kind of unclear exactly how that regulation and that governance should occur, but I want to stress that scientists are really taking this seriously. So scientists are really teaming up with ethicists and policymakers to try and figure out some of these sort of regulatory oversight systems that should exist. And one of the kind of examples I want to draw on here is the International Society for Stem Cell Research, sometimes called the ISSCR, which has come up with a lot of guidelines about embryo model research that are international. So all scientists across the world will try and adhere to these guidelines that are kind of quick to kind of come out, which means they can be really responsive to the pace of research, because of course that's happening far too quickly for primary legislation to keep up with. But the other really important voice in all of this discussion is all of you guys, right? What do we collectively think about these embryo models? Where do we feel like the lines should be drawn? And there are lots of kind of movements to try and get more public engagement, get more public dialogue. This was a dialogue event that was run a few years ago now. And you can see from these quotes that there's a real range of opinions, right? Some people think embryo models are completely different from embryos, they're just a bunch of cells. Other people think that because they have the potential to form something that's complex, they're really quite real, and therefore we should be kind of concerned and thinking about these structures. And of course, there's some people who felt in between. But I really would like to kind of encourage all of you here, now that you've taken this first step to come to this lecture, thank you so much, that you think about these questions and really participate in this discussion, because only by coming together and discussing these really important issues can we settle on something that we think will be useful moving into the future. And with that, I will stop talking and thank my lab, which is of course the most important slide. They do all of the hard work and the research upstairs in the laboratories. So thank you to all of them. Thank you for all of the support that we receive from various people around the building, our funders without whom we wouldn't be able to do our research. And thank you so much for coming. It's really nice to be able to talk to a room full of people. Thank you.